Good afternoon to all. Myself, Dr. Madhulika Gupta, Assistant Professor in the Department of Microbiology, Lady Brabourne College, Kolkata, would like to present my research work on the topic entitled Remediation of Textile Dyes, Direct Red and Acid Orange by Freshwater Cyanobacteria. Dyes can be said to be colored, ionizing aromatic organic compounds which shows an affinity towards the substrate to which it is being applied. Such substances are widely employed in the production of paints, textiles, printing inks, pharmaceuticals, food, cosmetics, plastics, photographic, and paper industries. Synthetic dyes are recalcitrant molecules that constitute the main residue found in the effluent of the textile dyeing industry. The US International Trade System classifies the dye into 12 types, such as direct dyes, bad dyes, sulfur dyes, organic pigments, reactive dyes, disperse dyes, acid dyes, azoic dyes, and basic dyes. The main steps of operations in a textile dyeing industry involve scouring, bleaching, dyeing, drying, and finishing. Many wastes are generated from each step like carboxymethyl cellulose, polyvinyl alcohol, caustic soda, mineral acids, sodium hypochlorite, hydrogen peroxide, heavy metals, different types of dyes, synthetic waxes, resins, etc. Thus, when these textile effluents are discharged into the environment, it causes certain direct consequences such as aesthetic issues, blockage of sunlight penetration through water, which diminishes photosynthetic activities of algae, damage flora and fauna of the ecosystem, groundwater pollution, depletion of dissolved oxygen of receiving water bodies. Indirect consequences being eutrophication, adverse effects on aquatic life, acceleration of genotoxicity and microtoxicity, suppression of human immune system, and causes of various human illnesses. The biological mode of treatment of textile effluent offers distinct advantages over conventional mode of treatment, like it is more economical, sludge accumulation is less, eco-friendly, converts dyes to simple inorganic compounds by mineralization which are less harmful to living forms. In this presentation, decolorization of textile dyes by freshwater cyanobacteria has been discussed. Cyanobacteria, commonly known as blue-green algae, photosynthetic pigments being chlorophyll A, phycocyanin, allophycocyanin, phycoerythrin, thylakoids occur singly, presence of polyhedral bodies, vesicles, fixed nitrogen by heterosis, thick gelatinous cell wall, it lacks flagella, nucleoid located centrally are some of the main features of this group. Examples being nostoc, anabina, limbia, gliotrichia, etc. Microbial decolorization occurs via two principal mechanisms, biosorption and enzymatic degradation, or a combination of both. Water sample was collected from a freshwater habitat. A number of genera such as Microcystis, Oscillatoria, Nostoc, Limbia, Gliotrichia, Merismopedia, along with some green algae and diatoms were observed in the crude water sample. The medium BG11 was inoculated and incubated under 16 hours of light and 8 hours of darkness in undisturbed condition for 14 to 21 days at room temperature. The isolated cyanobacteria showed the following features based on morphology, lactophenol cotton blue staining, and camera lucida drawings. Densely entangled blue-green filaments with colorless sheaths surrounded by gelatinous matrix, cross walls present, without heterocyst and akinites was observed. 10 to 50 mg per liter concentrations were prepared for each dye. The experimental setup consisted of sterile BG11, buffer, dye concentration, and inoculum, while the control setup contained all constituents except the inoculum. It was incubated for 8 days under the same conditions as mentioned. Absorbance was measured at 507 nanometer for direct red and 482 nanometer for acid orange spectrophotometrically to calculate the percentage of decolorization. effect of textile dyes on the morphology of the cyanobacteria was studied by microscopy and staining, while chlorophyll and carotenoid of the treated cyanobacteria was also estimated. Starch and NaCl was added to see whether there was any enhancement in the rate of decolorization. Finally, viability test was performed with ethidium bromide and acridine orange staining. So from the performed experiments, it was observed that isolated freshwater filamentous cyanobacteria was able to decolorize both textile dyes. There was a shift in the pH from 7 to 4.5. Chlorophyll was affected, but carotenoids severely affected by acid orange compared to direct red. Microscopic analysis showed that the filaments of cyanobacteria was intact, but sheath less dense at 10 mg per liter, while filaments were fully fragmented, decreased in number, along with disappearance of sheath occurred at 50 mg per liter after 10 days. Upon treatment with starch and NaCl, the rate of decolorization increased for both the dyes. Viability tests showed live green cells at 10 mg per liter and dead red cells at 50 mg per liter for direct red, and in case of acid orange, both live and dead cells were observed at 10 mg per liter and most cells dead at 50 mg per liter. Thus, it can be finally concluded that decolorization rate of direct red was more than that of acid orange, and this isolated cyanobacteria could be used as an effective economical alternative for degradation of respective textile dyes. I express my sincere thanks to the organizers of this international conference, RIAPS 2020, for providing me this platform to present my research work.